Okay, we're going to get started. Welcome. Thank you all for coming to our panel today. So we're going to be talking to you about the link between climate change and marine diseases and how that impacts both ocean and human health. I'm Emily Kunselman. I'm a PhD candidate at Scripps Institution of Oceanography.
I'm Kara Wiggin. I'm also a PhD candidate at Scripps Institution of Oceanography at UC San Diego. I'll start us off, if I can. Oh. So let's start off with a basic what are marine diseases, because I think when we say that, people kind of don't know the wide breadth of them. So it's just any disease found in the marine environment regardless of origin. So by regardless of origin, we mean, ones that are either from land that come from humans or ones that originate and are native to the marine environment. So these are ones that also can target human or marine organisms, and we're going to cover both of those today. So when we start talking about marine diseases, we want to look at them in the context of a marine disease triad. So these are just the points of each of these triangles are things that interact with each other to make kind of a complex system that all rely on each other to make sort of a disease ecology situation in the ocean. So we start out with environment at the top. So I'm going to go through just some of the environmental parameters that we focus on when we're looking at the ecology of a particular marine disease. So the main one really is temperature. And we see this on land too, that temperature can often have an impact on the diseases that we see. It's why we see so many more diseases in the global south and by the equatorial region. So temperature in the ocean is, for the most part, except for that little blue dot at the top, increasing. And this is a concern for diseases as they mainly are going to evolve and thrive in warmer temperatures. We then have salinity. This can affect things like where diseases travel, but also things like the health of a host. pH, which we generally see as going down and decreasing in the global ocean. And oxygen, another thing that can affect host health and can affect where pathogens live.
So now I'll move us on to the second point of our disease triad, which are the pathogens themselves. So a pathogen is a microorganism that can cause disease. This can be bacteria, viruses, protists. And what we see are that the impacts of climate change on the environment lead to alterations in our pathogen range, so where they're found geographically, and also their virulence, so how infectious they actually are to their host. And what is our host, our final point of our disease triad. And so this is the organism that is either directly affected by or carries the disease. And this may be us as humans, or it could be marine plants and animals. And again, the impacts of climate change on the environment can cause alterations in the host ranges, so animals moving to go to places where they are more physiologically adapted, and also it can cause alterations in host health. So the environment can stress out the host, it can put them in a more immunocompromised state, and really leave them far more susceptible to these pathogens. I'll turn it over to Kara to talk about her research, kind of focus on humans.
So today I'm going to talk about that first aspect of marine diseases, which are the ones that affect humans. So often when we think of marine diseases, we're thinking more from the animal side, but there's actually quite a few that can be incubated in the ocean that can affect humans when we interact with the water. So we often interact with the water through many forms. Recreation, so swimming, boating. If you have an open wound, things like this, you're often exposed to a lot of pathogens. Your occupation, so for example, fisher folk, anyone that's interacting with the ocean in any way is going to be exposed to these pathogens. And seafood consumption, which is our most common. That's the one you normally think of when you think of humans getting sick from marine diseases. Things like raw oysters, sushi, things like that. Hopefully you all have been spared, but likely not. So when we talk about human marine diseases, there actually are mostly, for the most part, derived from humans. So these are ones that can enter and persist in the ocean. And so oftentimes, they're being exposed through things like overboard sewage. So when we talk about large boats that are out at sea for a long time and they're dumping their sewage, so human waste into the water, that's any diseases we carry that can then live in the marine environment for extended periods of time. We see things like sewage runoff from rain and flooding. So when we think about climate change, we know these things are planning on increasing. Increased flooding in coastal areas, increased rain events, especially in places like Southern California where we're not used to these huge deluges of water. They can wash out sewage and other pathogens that have accumulated on time on land. And so this is why we see most of these diseases in these coastal areas where people are interacting with it quite a bit. Another kind of less commonly known exposure route of human diseases to the marine environment is from seafood processing. When things like infected handlers, that's often how we get seafood to be infected with diseases. Improper storage and transport of seafood, so if they're not kept at the right temperatures, those pathogens can really flourish. And cross-contamination between animals. So if you have multiple types of seafood in a market that are near each other and they're touching, those pathogens can be spread that way as well. So some examples of human-derived marine diseases are things like norovirus. If you have kids, you probably know this one. This is your classic intense stomach bug. This can travel and transmit between humans, but it also is very commonly received from seafood and can make it into the environment through sewage. We also have hepatitis A, which is another virus. This one used to be very common, but there's been a worldwide effort to vaccinate people against hepatitis A, so this has really gone down in the last few decades, which is great, but it's also still quite a threat. And then we have Salmonella and Shigella. These are two bacterial diseases. So you've probably heard of salmonella when people talk about salmon and fish and eating undercooked things. So those are the risks for that. These are mainly all sort of a food poisoning-like disease, but these can be fatal and quite often are if you're already immunocompromised, if you're young or very old. So then we move to marine-derived diseases. So those are the ones that kind of come from humans and enter the marine environment. But these are diseases that actually are native to the marine environment and just happen to be able to infect humans. So these ones, our exposure route is what I mentioned earlier. Seafood ingestion, opened wound exposure when you're swimming or recreating. And these ones are often not linked to wastewater and pollution like the ones I mentioned previously because they're just indigenous to the marine environment. They're growing in organisms. They thrive in the water column. And the most common example of this is Vibrio. So you've probably heard of cholera. Cholera is in the genus Vibrio. That's more of a freshwater one. So the saltwater ones and the marine-derived ones we're talking about are things like Vibrio perihemolyticus and Vibrio vulnificus. So these are probably words you haven't heard as often, but maybe you've heard the term vibriosis, which again is also sort of a food poisoning-like illness. And it's mainly from these two in the US, but there are a slew of other Vibrio. There's about 14 pathogenic Vibrios that are pathogenic in humans that we know of at this point. But there are about 30 species of Vibrio in the whole genus that thrive in the marine environment. So I'm going to start with a case study of the most common seafood-related outbreak bacteria. This is Vibrio perihemolyticus. Quite a mouthful. I've gotten really good at saying it very fast. So this is the one that globally is causing the most seafood outbreaks between all bacteria and viruses. So you probably hear about this when you hear about any seafood-related illness. This is mainly coming from oysters. So undercooked oysters, these harbor them very easily. And then the rest are coming from crustaceans, things like shrimp and other food like that that are undercooked. Now, Vibrio perihemolyticus is actually the disease that is the most of concern with changing climate change. This is a thermophilic bacteria, so it thrives in warmer waters, and we're seeing massive range expansion with Vibrio perihemolyticus and also range expansion of hosts. So you have a pathogen that's thriving in sort of the equatorial regions moving northward, and then we're seeing organisms moving southward that used to live in Arctic or in places like the Pacific Northwest, colder regions, moving southward. So you're seeing these organisms really interact where they never interacted before, and especially ones that we eat as seafood, where we never used to have to worry about these sorts of diseases in seafood, and now we're seeing it a lot more commonly. So these are really common, especially in the Pacific Northwest. We're seeing a lot of new outbreaks of these diseases, and it's causing a lot of disease in humans. So I just wanted to point out these two very small graphs, so sorry about that. The one on the top left, you don't really have to look at all the lines. The one I want you to focus on is the fact that over this decade, or so that's on this map, you can see that Vibrio perihemolyticus, which is the green line, is the only one that's increasing. So all of these other foodborne illnesses, which include things like listeria, salmonella, these ones are staying the same. So the Vibrio perihemolyticus and these sort of non-cholera Vibrio species are the ones that we're seeing already, these massive increases due to climate change, likely increased temperatures. And the graph on the bottom right, you hear it just shows the massive amounts of range expansion that we've seen from where it came from in the Indian Ocean. And over the last few decades, it's really spread across the world and is being incubated in a number of marine diseases or a number of marine organisms. So to do some shameless self-promotion, this is what I'm looking at as well. And it's something that I really wanted to point out because a lot of people really try to link marine diseases and pollution. And as they should. That's not saying that that's crazy. They absolutely should because they are linked with pollution. So one of the pollution aspects that I look at is plastic pollution. And a lot of people don't think that these two things are related, but plastic pollution is obviously so common in coastal areas, and it's really intersecting with the areas that these Vibrio species are thriving. So what we know about Vibrio perihemolyticus is that it is particle loving, which means that it always wants to attach to the surface of something. So you'll find it often on marine snow, the particles that float around in the ocean. And what we've kind of haven't thought about until recently, until I did it, is that plastic is a particle that's floating around in the environment, and it's probably harboring marine diseases. That was also a joke. There's plenty of other people that are studying this. So plastic pollution can potentially be a vector for this, and some other studies have shown that when you sample microplastics, that we're actually seeing a lot of virulent Vibrio species on these surfaces. So this comes in contact with humans if we clean it up, if we're touching it, if we're recreating, but it's also what happens when an organism is going to eat plastic? Is it going to move in there? So what we first need to understand is how Vibrio perihemolyticus is attaching to plastic. Is it growing rich biofilms? Is that changing when the climate is changing? So the main thing we're focusing on here is temperature. So we're trying to look at whether Vibrio perihemolyticus is growing different levels of biofilms or the amount of cells that's growing on the surface of a plastic when temperatures increase. So what we're finding here, okay, there's another thing. So what we're finding here is that when you look at the top graph here, just sort of orient you to it, this is the amount of biofilm that's growing on the surface of plastic. The blue boxes are a lower temperature, 18 Celsius, which is sort of like a standard spring-fall temperature in San Diego, and 25 C is about a high summer temperature that we're expecting to see a lot more often in San Diego. So we can see that there is a significant increase in the biofilm growth of Vibrio perihemolyticus on plastic when the temperature is increased, which means that we're probably going to see a lot more Vibrio being transported through coastal areas and being taken up by organisms as the climate change progresses. So what we know is that it's growing biofilm on plastic surfaces, it's increasing with temperature, and so we really have to pay attention to these effects of temperature on climate change when it's attached to a particle and also when it's in the water column. So, whoops, wrong way. So the second case study I want to do, Vibrio perihemolyticus is the most common globally found seafood poisoning. But the second most common one is norovirus. So this is not a bacteria, it's a viral outbreak. And about 21% of the outbreaks in the US are caused by viruses, and most of these are from norovirus. So we're seeing a lot of this change. This is also mostly from mollusks, things like oysters that are undercooked. But these ones, unlike Vibrio perihemolyticus, where we know there's a huge change with climate change, this one is more common during cooler months. This is probably not because of the physiology of the virus itself. It's probably just because it spreads between humans a lot easier in cooler months when we're in close quarters, and then it's going into the marine environment a lot faster. So we're just sort of seeing that like round table. What we are finding in the data is that norovirus is a lot more common recently, but we think it might just be due to the reporting aspect of it. We've gotten a lot better at detecting norovirus and figuring out that it is actually this species versus other seafood illnesses. And I just wanted to point out this great graph on the right here, because it's just really showing that, you know, your consumer, you get norovirus from your kids at school or wherever, you have it in your home, it's going into sewage. It's thriving in those estuarine and coastal areas, and then that's going into your seafood and going right back. So we're just sort of seeing this cycle between humans and seafood and disease that I think a lot of people don't think about, and it's pretty gross. So now you're asking me, okay, this has all been horrifying. How many people are actually impacted by this? And this is the one number I can find. I've scoured the internet, and the number I can find is that there's 100 million enteric and respiratory illnesses worldwide from swimming and consuming shellfish. So all of the sort of interactions with the ocean, this is the number we have. This is a lot. But what I really want to point out is that truly this is the only number I can find. These things are not really reported on. They're not observed. And so one of the things that I really want to stress today is that we need so much better reporting on these marine disease systems. If we're planning on seeing these things increase, we need records of it. And in the US, we have a foodborne disease outbreak surveillance system. But what I want to point out with that name is that it only focuses on outbreaks. That means it's only when a group of people are getting it. So if maybe one shellfish farm or something has a big outbreak, that's going to be the only thing that's reported. And so we don't have those individual cases where people are eating randomly getting reported. So the number of illnesses is probably highly underestimated. And so we have really no clue what the true impact of marine diseases on humans are right now. And also the system is passive, so it relies on voluntary reporting. There aren't people going out and trying to collect these numbers. This is just up to healthcare workers to report these outbreaks to the system. So again, really underestimating these illnesses. Another reason that they're underestimated is because these illnesses are usually self-limiting, which means that you just sort of heal on your own. You don't need to usually go to the hospital. You could just stay home and deal with it, maybe go to the drugstore. So these things often aren't reported to healthcare systems if you don't need to actually be hospitalized. And usually you're hospitalized and they're reported when you're immunocompromised or young or old and have a severe illness, which is not usually the total cases. So we also struggle with the amount of testing that we do, especially in areas outside the US. These testings they're kind of expensive, they take time. Hospitals sometimes aren't going to test for the etiological agent or which bacteria or virus is causing the disease if it's not worth it. If you just need an IV bag and you're gonna be fine. So reporting isn't uniform. We need more of it. We need reporting that is not just outbreak specific so that we can truly understand the impacts of climate change on marine diseases and on human health. So I'm going to turn it over to Emily, which is going to go for the marine organism side of this question.
Thank you, Kara. That was really, really a good overview of how these marine diseases are going to affect humans. But I want to start to tell you why you should care also about how they affect marine organisms themselves. Primarily for me, I see it as a source of food. Actually, 17% of animal protein that we consume worldwide is from seafood, so fish and shellfish. And I mean, there are plenty of other reasons that marine organisms can produce medicinal compounds for us. They can produce oxygen, cycle nutrients, protect our shorelines from storm surge. They can purify water through the many organisms that do filtration. And they provide great habitat for recreation and tourism and important for cultural traditions. And so these benefits are at risk with climate change. We know that the ocean is warming. We can see in this plot the ocean heat content has steadily increased since about 1970. They may be a little small for you, those numbers, but that start of the incline is about 1970. And we know that these increased water temperatures are linked to the emergence of new diseases. And like I said earlier, ocean warming and some of these impacts that climate change can have on the environment are leading to the expansion of the range that the pathogen can hold. This can cause them to overlap more with other marine organisms, and it can make them more infective to their host. And these diseases can kill marine organisms in addition to just making them sick, which really can threaten farmers and their livelihood, but also threatens us because a lot of people have been mentioning that the ocean is the future of our global food system, and we really don't want that to be at risk. Also, this puts endangered species restoration projects at risk. So we don't want to try to curate this habitat for species, put them out there, and then just have them be susceptible and fail due to diseases. I'm a scientist, so I don't feel like I can just tell you that marine organism diseases are increasing with climate change. So I have a few examples for you. The first one is coral. And I wanted to talk to you about black band disease, which is actually the first disease that was ever categorized in corals. And this is a very interesting disease because it's not just caused by one pathogen, it's caused by a consortium or a group of different types of bacteria. And you can see in the photo that there's this black band surrounding the white. The white is actually coral skeleton, so that mat has laced the tissue underneath, and it continues to spread throughout the coral. And we see that this is more prevalent with increasing temperature and decreasing water quality. And this has allowed for black band disease to increase the number of different coral species that it can infect and the geographic range that it persists in addition to just how the global warming impacts the pathogen and the pathogens, I guess, the group of bacteria, it also leads to stress of the host. So when organisms are stressed, they don't have the same capability of launching an immune response and controlling these bacterial populations like we do normally with all of the microbes around us. The second example is with seagrass. So seagrass are really, really important for oxygen production, for habitat structure. And we see that with seagrass wasting disease, which is caused by a protist. You can see that brown area in the photo. This disease has reduced prevalence in cooler waters, so in the deeper waters that we find these seagrasses in. And if we have warm water starting to intrude into those spaces, then not only are the seagrasses closer to shore at risk, but now the ones that are deeper and perhaps more resilient are actually also going to be affected. Wrong way. So also with fish, there's this disease called ichthyophoniasis, quite a mouthful. I practiced that multiple times. So this parasite can cause lesions on the fish. You can see in these photos a bleeding ulcer in the top left-ish quadrant. In the top right, it's pointing to a fish lesion, but it's very hard to see. And then they also develop something called sandpaper skin, which is in the bottom right. And this really renders the fish unmarketable because you don't want to pick up this fish in the market and you don't want a fish that has a parasite on it. And we've seen that infection and mortality has increased with increasing water temperatures. This is actually something that really needs to be studied more on an oceanographic scale to see how it's actually impacting these fish populations in total, but it's definitely an area of concern. And now to my priority, which are marine bivalves. So I study abalone withering syndrome. This is caused by a bacteria that is intracellular, so inside cells. And white and black abalone are both endangered species along the coast of California that are very susceptible to this disease. There are many other abalone that are also susceptible, like red abalone, which are actually cultivated in California. And this disease attacks the digestive tissue of the abalone, which prevents it from actually absorbing nutrients from its food, and so it actually starves to death, which is really, really sad. Especially if you see one of these abalones and their little eyes, it's really sad. So they have a scoring system for this disease, which you can see on the bottom. At 0, that's a healthy abalone. We call that its foot, its tissue. It has a shell on top and that tissue on the bottom. And as you see, when we progress from 1 to 2 to 3, we see the shrinking and really the decomposition of that tissue. And these clinical signs of wasting and also of eventual mortality have been linked to seasonally warm temperatures. So in the same case as the seagrasses, efforts to outplant these abalone are focused on deeper, cooler regions because we know that they will be less susceptible to this disease there. But if we have intrusion of warmer water into those spaces, then that really doesn't bode well for these efforts that we're trying to do. And there's not enough white abalone in the wild to reproduce on their own right now, which is why they're so critically endangered. And then my other favorite marine bivalve is the oyster. I study the Pacific oyster, which is cultivated worldwide. It's really commercialized and very popular. And I study the Ostrid herpes virus. So yes, I study oyster herpes. And this virus is not able to kill oysters below 15 degrees Celsius. And we've seen this around the world. I've conducted my own experiments that I'll show you. And I was in France where they were studying it, and they're just unable to infect and kill organisms below this temperature. But with increasing temperature, mortality is also increasing. So this photo is of an oyster that I unfortunately killed from exposing it to this virus after just 2 days. And so you can see that the two shells are gaping. This is how we know it's dead. The adductor muscle that holds those two shells together cannot properly do so. And the other interesting thing about this virus is that we even detected it for the first time in 2018 when we had a major marine heat wave that year in San Diego. And this led to 99% mortality in the farm where it was detected. So we're trying to start these aquaculture operations in the San Diego Bay, and this really put us back by I think at least 5 years now. So I wanted to demonstrate just how widely studied this disease, how widely studied temperature, the interaction of temperature in this disease are. So these are just a selection of papers that focus just on temperature and OSHV1 from France, from Australia, from the US, and also me, I study it. So I just wanted to walk you through a little experiment that I did to demonstrate this impact. So we take a homogenate of this virus and infect it into oysters. And then we put those oysters in seawater, and they're able to replicate that virus and then expel it into the seawater. And we use that seawater as a method of natural infection of the oysters, as if it was happening in the wild. And so we have a control group, because we're great scientists, and a virally exposed group. And I did this at 4 different temperatures, 15, 18, 21, and 24 degrees Celsius, which is about the range that you might expect in San Diego Bay, where this virus is present. And we monitor the oysters every day to see how many of them died. And I just want to orient you to this plot a little bit because it's beautiful, but may need some orienting. So on the x-axis horizontally, you see the time in days. So like I said, every day we were monitoring, taking a time point, and it goes up to 16 days after we exposed them to that seawater. And then on the y-axis, you see oyster survival. So as it goes down to below about 0.25, that's less than 25% survival. You might notice that at 15 degrees Celsius, which is in the green, none of the oysters died. So again, showing this phenomenon that the oysters cannot be killed by this virus when the temperatures are that low. But as you work your way up to 18 degrees Celsius and higher, we see that over 75% of the oysters are eventually dying. Now, we have a p-value of less than 0.0001, which is a statistical test that sees whether the temperature causes differential mortality. And we do see that. So at 18 degrees Celsius, it actually takes longer for the oysters to start dying, whereas at 21 and 24 degrees Celsius, they start dying within 1 to 3 days. So this is really rapid, and this is really a concern for aquaculture, because if we detect it, it's probably already too late. It takes us probably that amount of time to run our assay to test it. And so this is really concerning because, like I said, this is the range that we see in San Diego Bay every year. About half the year, it's over 18 degrees Celsius. And so the oysters are susceptible to it every year, and we need to understand what else is triggering them in addition to this temperature. So I'm going to wrap up conclusions from both Kara and I's talks so far. So hopefully we've convinced you by now that marine diseases are increasing with ocean warming, and this is going to lead to increased human illnesses, a loss of our harvest and potential aquaculture operations, and potential failure of species conservation projects. This is because new diseases will emerge, the range of our current diseases will expand, and the range of hosts will change, both factors that cause more overlap between disease and the hosts. And our host health will be stressed out by this ocean warming, which leaves them more vulnerable to these diseases. I'm going to turn it back over to Kara to present to you some of our recommendations based on our learnings, and then at the end of this, we'll have a Q&A, so get your questions ready.
Amazing stuff. Really, really concerning for marine diseases in marine organisms. So since we're here at CUP, let me give you our policy recommendations. So we wanted to sort of highlight this section of the Ocean Pavilion's COP28 declaration because although we've heard a lot, especially from some people in the audience, about how important observing capabilities are for climate and our climate, physical variables, it's also important to do some large-scale ocean observations of our biological variables, and especially for marine diseases. And this will help us understand the impacts of climate change on the ocean. So the first thing we need to do is really work to establish long-term monitoring programs in specifically coastal regions where people are highly populated. And this will help us prepare for diseases. We can figure out where the diseases are happening. We can relate it to physical parameters in the ocean like temperature, salinity, pH, and oxygen. And this will help us figure out which diseases are out there, where they're moving, and which seasons they're the most prevalent, so that we can inform our fisheries and our aquacultures. We can inform people of when it's the safest to eat seafood, among many other things. We also really need to address policy to address current and future marine wildlife disease management. There's been a few bills that have gone into Congress and have not gone through and not passed. And these things will just allocate money towards disease, addressing a disease that's currently happening. If we're seeing a very large outbreak in people, if we're seeing a very large outbreak in an aquaculture facility, this can be money that is allocated to deal with that when it's happening, when we have these emergencies, so that we can deal with them. And it also can help us prevent future marine wildlife disease through all the things we're talking about throughout everything at COP, reducing emissions, reducing pollution, sort of dealing with these parameters that can exacerbate diseases in the marine environment. And on my own thing, as I mentioned earlier, we really need to improve data reporting. We also really need to improve data reporting for marine diseases as well. We see this all over the place, that farms are telling us, personally, that they've had disease outbreaks, but we're not seeing that in the literature, in government reports. But especially for human waterborne illness, it's so underreported, and the numbers that we have, I can tell, are just insanely low. And so we really need better data reporting for all of this, so that we can really understand this unappreciated aspect of climate change on human and environmental health. Oops, keep going the wrong way. And so kind of moving from policy, how can we have research to sort of address these goals? So we really need to increase research for marine disease systems. The more I've gone into this field in the last handful of years, the more I realize how little people are looking at this and how little we know about the whole marine disease system. So I mentioned that marine disease triad. It's so hard for us to know the information about every point of that triad for most of the marine diseases we have. The two I mentioned are really like the only ones we have much information for. We don't know most of the hosts that most of these diseases infect. We don't know the impacts of different environmental parameters on host health, on pathogen virulence, on these sorts of things, and how all of these things are interacting. The data is so little, and we need so much more to really understand and prepare for the future of this reality. We also need to understand the underlying mechanism of how climate change affects disease. As I mentioned, we need to understand those physical parameter effects. That's something, we often just study how the disease affects the host, but how does temperature change it? How does pH change it? Things like that. We also really need to analyze the clinical data. So as I said, we need so much more reporting of it, but then we need someone to go through that and then tell us what it means, relate it to ocean health, relate it to climate change, and get that information to policymakers so that it can really be implemented. And then we really need to take up a notch our monitoring data. We need to use models to really understand where climate change is enhancing, and then how that interacts with where diseases are going, where hosts are going, how those things are going to interact. I'll move over to Emily to finish up on our environment recommendations.
Yes, so primarily with the environment, we just need to make sure that we're constantly improving marine ecosystem health and not damaging it like we have probably a lot in the past. One of those ways is through reduction of point and non-point source pollution, which exacerbate these diseases because it's an additional stressor, a multi-stressor situation. We also need sustainable aquaculture practices. I'm not a pessimist, we can manage these diseases, and I've worked with people at the Department of Fish and Wildlife in California and with the Port of San Diego, and they're really, I think, doing a great job of managing this disease so that we can still grow oysters, like a really important, really sustainable seafood, while mitigating the effects that it has. So not moving oysters too much from place to place. We really need local, small-scale, circular economy practices. And finally, we just need to enhance biodiversity. This is just across the board with everything. If you have a diverse community, multiple species, then you're going to be more resilient against these diseases. You're more likely to have a few organisms that survive and maintain that population. So thank you so much for listening to us. Kara and I really love talking about this topic. You can take down our email and follow up with us. You can follow us on Twitter/X, and that's our LinkedIn profile as well. We love using LinkedIn. And now we're going to move into the Q&A portion of this talk. So we'll sit right up here and answer anything.
Hello, everyone. Feel free to ask a question. And if not, I'm going to ask Emily a bunch of questions. Yeah.
The microphone?
Do you have the mic?
Hi, Grace Colley from Scripps Oceanography. I was wondering, you're talking about these diseases and you're talking about how they're influenced with climate change, but as we're at COP, and the reality of the situation is we're not going to get emissions under control in time for maybe like the next 5 years. So how do you manage diseases in that time, and is it like UVing wastewater systems? Is it adding antibiotics? How do you go on the defense until we can get the climate and the heat under control that's really expanding their range?
Do you want to start?
I can start.
Okay.
So I do think, number one, that oysters are highly resilient. I believe in them. I believe they can do it. And as I mentioned kind of in the last slide, like, we can manage these diseases in aquaculture. We can make sure that our growing seasons line up with when animals are not going to be susceptible to the disease. A big issue with globalization is that we've moved these diseases all around the globe. And so if we are going to be facing warmer temperatures where these pathogens are likely to spread rapidly, then we just need to make sure that we're focusing things locally, which is really great, because then we're also reducing carbon emissions by not shipping and not flying things. And ideally, you can just walk to your closest oyster farm. That's how I would love the future to be. I think on the other vein, like with conservation projects, because I study this endangered white abalone species, this is concerning. I don't think that I have an answer for you right now of how we can really mitigate it, other than just across the board, we need to keep doing more research, and we need to assess how these animals do when we put them out and when we outplant them. And we just need to do things instead of being like, oh, well, this needs to be perfect. We just need to try things and see how they work and do it responsibly, and then assess how it goes.
I think on that note, when we're talking about solutions, Emily and I just wrote a big paper about potential microbial interventions on aquaculture, and sort of using those to mitigate the microbial diseases. And so things like antibiotics, it's a solution. It's not the best solution, because antibiotic resistance is a really big issue in coastal regions. But these are tools that can be used. There's also things like phage therapy and using phages to eliminate diseases. But these things require a lot of research and FDA approval and a number of other permitting and other issues with these solutions. There's also, you mentioned UVing water, an option, an expensive option, but there's like a lot of solutions in sort of the aquaculture space that we can do to sort of prevent diseases. There's been a ton of research looking in the Vibrio world on how we can prevent, storage and handling of seafood to prevent the proliferation of the diseases as they're being shipped or as they're like waiting in a grocery store, which really includes sort of depuration, putting them in clean water, keeping them at low temperatures and things like that. So there are sort of a lot of like technological solutions that we can use. And really it's sort of like the cheesecloth COVID thing where, not everyone is going to be a silver bullet, but if you use so many of them, hopefully they'll overlap, and then you can sort of reduce those marine diseases. So we just sort of have to use these technological innovations in the meantime to manage diseases, but it's really hard in the wild. These are only situations where we can control, but in the wild, it's going to be things like Emily said, like preventing the movement of animals between areas, preventing ballast water. I know a lot of places require the testing of ballast water now. That's going to be really important for moving diseases between continents. Things like, not importing marine animals between different areas, things like that. So that's really the only interventions we can really take on managing marine diseases in the true wild. But then there's also things like Emily mentioned, reducing pollution, things like that. That's going to deal with it from the human perspective. So yeah, just managing those multi-stressor effects, not trying to stress out the host so that when a disease happens, they're a little bit more resilient. In those types of interventions, but it's tricky. These things are, as we mentioned with the triad, like it's a really complicated and circular system where these three pieces are all, having their own impacts. The hosts have their whole slew of things that make them either susceptible or resilient to disease. The pathogens are changing, the environment's changing, so it's really quite a complex system.
Yeah, I emphasize complex. I wanted to add too, when you mentioned we have technology, I also want to mention we do breed resistance in animals against these diseases. We can kind of try to speed up evolution, but bacteria and viruses evolve very, very rapidly. So
also things that require research and money.
Exactly, require a lot of money. We need more resistance breeding programs and genomic tools to be used in them.
Sweet. All right, this is working. So I have a couple questions. The first one is to you specifically. It's a bit more technical. And the second one is very broad. So my first question is, so the Vibrio thing I can't pronounce. Yeah. So which strain is most virulent, and was there a more significant difference in biofilm formation due to like temperature differences?
Yeah. This is a great question. Love it. So basically, so I measured 8 strains in those experiments, but there's thousands of strains. There's so many different strains. And what I wanted to highlight between strain differences is 2 things. One, there's sort of clinical strains, which are the ones we know can infect humans. And then there are environmental strains of Vibrio parahemolyticus that don't infect humans. So even though it's a Vibrio that generally can, there are strains that are just sort of hanging out in oysters and they're totally fine. I don't see any significant differences between clinical and environmental strains in their biofilm formation, which is not super surprising. And other studies have found the same when they're looking at biofilms on other types of objects. But it does sort of make it so that we can't just like say, oh, for sure the clinical ones are growing these huge biofilms, and that's the big problem. But in the strain section is that there's also pandemic strains, which are the ones that are sort of like super clinical, and they can really spread between humans. They can spread really fast between organisms, and these are quite concerning. We had a pandemic strain breakout in Washington a few years ago. There's been a lot of pandemic strains found in Japan and China, where Vibrio is really common there. So, we have to keep an eye on those pandemic strains that really have the potential to wreak a lot of havoc. But for the most part, those aren't what's mainly circulating. They're just sort of popping up. So, not a huge amount of difference in biofilm, but potentially still is going to cause differences down the line.
For sure. Thanks. So my question for both of you then, my second one, is, in your opinion, how big of a role does investing in clinical research for marine-related diseases have in building climate resilience?
Clinical disease?
Yeah. I mean, so, yeah. I mean, just like... No, you go first. I need to think about it.
So just to clarify, you're saying investing in clinical like human disease and treatment and reporting? Oh yeah. I guess I'm not in the human health world as much, so I don't know as much about how how much money they have or don't have. But in any case, yes, these diseases are going to impact us, and so having more funding to places that are studying the diseases and the impacts on us and how to treat them with really innovative solutions, I think. So Kara kind of mentioned phage therapy and probiotics and moving away from antibiotics. I think that's our future. So that's one thing that I would say I think more research should go towards that.
Yeah, so now that I have my thoughts. So basically, with clinical treatments for humans for these diseases, there's not a whole lot. And, especially with things like norovirus, when you read about it, the main one we've had success with is hepatitis A. And we've had a global vaccine campaign to get that. We're seeing levels of hepatitis A go down. But truly with the Vibrios, there's not much we can do with bacteria other than antibiotics and phage therapy. But phage therapy is in its crazy infancy, and it's really very not approved, nor is it studied where it needs to be yet. People are working on it, but it has a lot more work to do. The other issue with phage therapy is that it's often really strain specific. So it's really hard to, all the literature I've read is that we're having a hard time finding a general Vibrio parahemolyticus phage. And they adapt very fast. So, these clinical treatments are really tricky to develop. It just sort of remains to be seen what people are going to be able to do to treat that. It's really more about sort of maintaining health, because we see more increased mortality with things when people are immunocompromised or young or old. So, increasing health. But then, it's also knowing that if you're young or old or immunocompromised, staying away from undercooked seafood. So, like, clinical disease is going to be in the sort of prevention side. Will be where the biggest fund should go. Really is public education. But there's also risk to public education. Anytime I tell people my research, they're like, "Oh my God, I should not eat oysters anymore." And it's like, no. Like, we want to encourage, healthy, sustainable forms of aquaculture and seafood, like ocean food, because this is definitely going to be part of the future. So we don't want to take people away from it. It's just education, proper food handling, proper, eating them at the right times of year, preparation, maybe cooking them more, things like that. So, but it's a really good question, because there's a lot of multifaceted issues with it in terms of prevention, overall human health, and our sort of biological interventions. So thank you for the question. That was great.
Hi, Mitchell Chandler, Scripps Oceanography. What is a phage and phage therapy?
Great question. So a phage is sort of like a virus. And it's something that can infect a bacteria, and then it will kill the bacteria. So phages are the way that they live is sort of like going into a bacteria. It sort of embeds itself in their genome in a way, and it's just sort of lyses them. And so they're so prolific in the ocean. Phages are all over the place, and it's sort of how bacteria are managed. And a lot of times when we see big bacteria outbreaks, the phages come in, wipe it all out. So they're all over the place. They're really, really common, and it's a way we can use to manage bacteria. But they do tend to be pretty specific towards a strain.
Yeah, I'll elaborate to give you a little bit more information. These phages, like Kara said, there are a lot of them. They can be very bacteria specific. But they also are at a constant, we call it the arms race between bacteria and viruses. So a virus is constantly evolving to infect a bacteria, and then a bacteria is evolving to be resistant against it. And so one of the main difficulties with phage therapy is that you're going to need to have constant development of the evolved, phage against that bacteria. It's just going to be a totally different structure of how we use phage therapy to treat diseases. We actually have a really cool success story, though, from a faculty at UCSD who used a phage to treat her husband's brain disease, and he's still alive, and I saw him 2 weeks ago at a Christmas party.
She has an amazing TED Talk. Highly recommend.
Stephanie Strathdy.
Yeah. Really just promoting other people. Question in the back. Oh, sorry.
Michael Dotzel, Woods Hole Oceanographic. So very, very interesting talk, ladies. Thank you very much. So I'm definitely a neophyte when it comes to microbial work or microbiomes. I'm sort of curious. So I'm wondering these bacteria or these viruses, they kind of parasitize these oysters and other different kinds of specific species, but they also kind of use plastics and particles as substrates, right, as you found. And my question is, in the same way that plastic has been demonstrated to bioaccumulate in organisms, to what extent do you expect that these viruses or associated bacterium or something also bioaccumulate in other organisms that are higher up in the food chain that we eat, aside from just oysters and other organisms that specifically you've demonstrated as examples?
Do you have an answer?
I have, I guess, a quick thought that I had. So the thing about when I tell people I study oyster herpes virus, they're like, oh, like, should I not eat that oyster? I'm like, number one, it's going to be dead. You're not going to get that virus. And number two, those viruses are very host specific. Once it's evolved to affect an oyster, it's just going to affect an oyster. So maybe in that case, we're not really going to worry about, I guess, bioaccumulation up the food chain. But bacteria-wise, Kara talked a lot about Vibrio. I didn't even mention it, but Vibrio also infect a plethora of marine organisms, oysters being a huge target, especially in hatcheries, which are a little more controllable, that we can manage a little bit better. When an oyster gets sick with this virus, there may be opportunistic Vibrio that also infect the oyster. And then when that oyster dies, those bacteria are leached into the immediate environment, and those can definitely go on to affect other marine organisms. So I guess more of like a horizontal transfer, but it is a concern, I think, with Vibrio and then potentially other bacterial pathogens that we don't have very well described yet.
Yeah, and in terms of like a true bioaccumulation situation up the food chain with Perihemologous, I haven't read any studies that are looking at that. I haven't read every paper, so that it could be out there, but I haven't seen it. And so, there's also the question of how long is the Vibrio sort of living in, it's definitely alive and thriving in guts of organisms, but then if it's getting eaten by another organism, is it staying in there? So yeah, really, honestly, my answer is, I don't know, but probably.
Hi, thank you for that talk. I'm Monica from Scripps, and I wanted to ask you, so you described your finding that Vibrio is accumulating on plastics in the ocean. And I was curious if you think that this work can, inform policies to reduce, our trash dumping in the ocean, like from ships, from Navy, for example, just locally to San Diego, and how this work can be translated into actions that way.
Yeah, this is a really good question because I love to talk about plastic pollution. So when we talk about plastic pollution, there are so many ways in which it's damaging, and it's human health, environmental health, entanglement, toxicity, things like that. But so I look at this as sort of just another example of the ways that plastic pollution is wreaking havoc on the environment, is that it's accumulating pathogens that's potentially damaging to humans and marine animals. And so it definitely is part of the weight of evidence for the damage that plastic pollution has on the environment, which is why it's sort of a compelling concept for research. And one of the things that I immediately thought of when you asked that question was, I don't know if you guys recently heard this past year, there was a huge sargassum wave that came into Florida, and sargassum tends to accumulate a bunch of other stuff. And so the sargassum, blooms in the center of the ocean, they tend to accumulate a lot of plastic. So when that when it washed up, it sort of washed up a ton of plastic pollution from the open ocean as well. And they were finding a ton of Vibrio on those plastics. And so, you have to be careful when you're touching that. And so these are just all big pieces of evidence for the reduction of plastic in the marine environment, because we always just were like, plastic is dangerous to whales and things that eat it, but here's another aspect that people haven't thought of as well. So definitely advocating for the reduce of plastic emissions into the marine environment. Thanks for that question. Any other questions? Okay.
Hi, this is Lynn from Scripps. This is fascinating. So as the oceans change temperatures and even a natural climate change, and then because that's been going on, these organisms have been sort of dealing with changes for all of their evolution. Is it disease or the organism's metabolism itself that changes their ranges?
This is a really good question. So this is one reason why one of our points was that we really need to study the underlying mechanisms of these diseases, because there are a lot of cases where we know just a little bit about how it affects the organism, but not everything. But metabolism, I've learned so far, is a huge player in this. So another component they've studied with the OSHV1 virus is actually food. If you feed the oysters, then they're even more susceptible to getting the virus and dying, as opposed to if you starve them ahead of time. And so they think that this is related to the metabolism. If the oyster is metabolizing faster, then the virus is more able to replicate faster and overcome the oyster's immune system response. So yeah, I think that's the main response that I have there.
Yeah, so definitely, and in terms, something that we sort of mentioned was the issue with change with temperature is that the range expansion of the organisms is changing. So we see this a lot with crabs, but we're seeing it also with oysters as well. And it's the oysters are struggling to survive in hotter temperatures, so they're moving northward. Not on their own 2 feet, but their range is expanding. And with metabolism, either their metabolism is faster and viruses are thriving, or it's that, something that we see a lot is that when they're exposed to warmer temperatures and they haven't been able to sort of expand their range, they're just a lot more susceptible. Their immune system is either overworked or it's sort of like turned off in a way. And so their health is diminished. And then that can really, a disease that never used to bother them will now infect them. And then we're also seeing pathogen range expansion as well. They're moving really quickly into environments that they never were. So you're seeing the intersection between organisms that never saw these pathogens, and now they're seeing them. So yeah, there's a lot about, the host health side of it that we really don't know, which is what Emily was trying to say. We don't really know a ton about how changes in host health are making them most susceptible to different pathogens, and then the effect of the environment on that. Temperature stress, oxygen stress is a big one I read about a lot. Stress from pH. pH is big for shell degradation, but it can also just affect, salt transport and a lot of other physiological aspects. So all these things that we really don't have a lot of data on the underlying impacts of these physical changes in water on host health, and then how that translates to pathogen.
Yeah. I also wanted to make sure I touched on the pathogen side, because we also don't fully understand, maybe at a cellular level, how the virus is infecting the oyster, how a bacteria is infecting an abalone, in relation also to temperature. What is it that the temperature is doing to allow the pathogen to get into the cell or the virus to replicate? Like we don't know these things, and I think it's hard for me because I want to really understand these things and I don't always have the correct technology to approach that question.
Did we answer your question okay? Yes. Okay, good. Just want to make sure we covered it. Any other questions?
We might have time for like one more question if there is another one from the audience.
3 minutes. And if there's one, perfect. Yes, please.
This is kind of directed at Emily, but Kara, feel free to chime in. I feel like I've heard a lot about when you congregate a lot of similar organisms. So when in the aquaculture idea, that disease spread more prevalently. How does aquaculture deal with that? And also with humans trying to live in more, close spaces, especially around marine areas because it's beautiful. How is the congregation of all these organisms spreading it?
That's a really good question. So a lot of this is due to density in aquaculture. Fish pens off the coast, you see more diseases. The oysters we had in that farm in San Diego Bay, they were the ones that were affected, but we haven't actually detected it or seen, associated die-offs with the virus from wild oysters. And so I think that's a problem that, of course, we would want to overcome. We want there to be less dense growing baskets, but there's not a very big profit margin in some of these shellfish aquaculture industries, and it's very hard to permit and license. It's very cost and time intensive, and so it's really difficult to get a bigger plot to grow the same amount of oysters. Why would you do that? So I think it's definitely a problem, the density aspect of it. But interestingly, I will note I was reading about the seagrass wasting disease a little bit more, and they found actually that with higher density, the seagrass was more resilient against seagrass wasting. I was really surprised because you normally don't see that happen. And I think they attribute it to, if there's more seagrass, it's a healthier seagrass patch as opposed to patchy seagrass beds that apparently they're not as healthy. So I thought that was interesting. We just need to know case by case what it is. And I guess I would love to see subsidies for like shellfish and bivalve aquaculture because it's difficult.
Yeah, and I really want to highlight that excellent point you made about that it's case by case because, and that's what we're trying to say is that, you know, these things are going to be disease specific and host specific. So we can't make these general policy changes on just, oh, reduce density, oh, reduce, you know, interactions or whatever. This is going to be disease based, which makes it all more expensive and complicated, but that's really how we're going to deal with it is really by going by individual diseases and figuring out the changes that need to be made for each one as we deal with it.
So I think that's all we have time for. Really happy that you all came and attended today. Like I said, Karen, I love talking about this. So if
Please
you want to
ask
come
us
up
other questions
and ask us more, we're happy.
Yeah.
Happy to chat.
Thank you so much for being here.